Good afternoon and welcome, everyone. Welcome back, those of you who were with us yesterday, and welcome to our faculty and students joining us today. It's an honor to see so many of you join us in celebrating the life and legacy of Professor Daniel I.C. Wong, Institute Professor of Chemical Engineering. I won't attempt to repeat all of my introduction yesterday, but we'll say just a few words to set the stage for today's programs. As we all know, Danny was one of the founding fathers in the discipline of biochemical engineering. His work in bioprocessing, enzyme technology, and mammalian cell culture set the stage for the modern biotechnology industry and for many of the advanced pharmaceuticals that are saving lives, even as we speak. Yesterday, we heard reflections from Danny's students about his gifts as a teacher and a mentor. Today, we will be focusing on his legacy as a trailblazing scholar. This morning, we will be hearing from 4 current leaders in the fields of biochemical engineering and biotechnology, who will be talking about the state of the discipline and Danny's legacy in it. Then, following lunch, we will hold the annual Daniel I.C. Wong Frontiers of Biotechnology Lecture, which this year features Dr. Melissa Moore, Chief Scientific Officer, Platform Research. Dr. Moore is responsible for leading Moderna's research in mRNA biology, delivery, and computation science. Following her talk, we will close out the afternoon with a series of short talks by industry leaders. These talks will touch on their current work, as well as the questions that they think are most exciting looking to the future. Danny was forever at the forefront of the discipline, and through these talks, we might imagine what he would have been excited about and working on today. With that, I will introduce the morning speakers. This morning, we will be hearing from Professors Phil Sharp, Charlie Cooney, Crystal Prather, and we'll finish by hearing from Dr. Nubar Afeyan. I'm going to introduce Dr. Sharp now. Please allow me to introduce my colleague Phil Sharp, Institute Professor in Department of Biology. Phil completed his PhD at Illinois Urbana-Champaign and his postdoctoral work at Caltech studying the molecular biology of plasmids from bacteria in Professor Norman Davidson's lab. In 1974, he accepted an offer by Salvatore Luria to come to MIT. Phil's research interests have centered on the molecular biology of gene expression relevant to cancer and the mechanisms of RNA splicing. Much of his work has been conducted at MIT's Center for Cancer Research, now the Koch Institute of Integrative Cancer Research, which he joined in 1974 and directed from 1985 to 1991. One of his most recognized advances was the discovery of RNA splicing in 1977. This work provided one of the first indications of the startling phenomena of discontinuous genes in mammalian cells. The discovery that genes contain nonsense segments that are edited out by cells in the course of utilizing genetic information is important in understanding the genetic causes of cancer and other diseases. This discovery, which fundamentally changed scientists' understanding of the structure of genes, earned Phil the 1993 Nobel Prize in Physiology or Medicine. He has also been the recipient of many awards and honorary degrees over the course of his incredible career. In 1978, Dr. Sharp co-founded Biogen, now Biogen IDEC, and in 2002, he co-founded Alnylam Pharmaceuticals, an early-stage therapeutics company. His work also brought him into frequent collaboration with Danny Wong, as I know he'll discuss today. Please join me in welcoming Professor Phil Sharp.
Thanks, Paula. It's an honor to be invited to participate in this very touching and interesting symposium remembering Danny Wong's contributions. Danny came to MIT 7 years before I did, in 1967, and was a very established professor. And what I'm going to talk about today is not Danny and his unbelievable set of charms and interests, his love of Chinese food, his wit and charisma, his can-do attitude, his kiss, keep it simple, stupid. But I'm going to talk about his role in changing biology and biotechnology at MIT and across the world. Danny lived in interesting times. And that's what you could say about the 70s and the 80s. And from a biological perspective, what happened was the confluence of three technologies: recombinant DNA, where you could combine DNA, DNA sequencing, where you could actually tell what DNA you were using or had, and chemical synthesis of DNA. And that created synthetic biology. For the first time in the history of man, we were able to synthesize organisms in theory. And we've been working with that ever since. At the same time, there was a number of discoveries as this technology began to be used in the analysis of human cells and having great implications for biomedical science and biomedical therapeutics. And one has been mentioned, and that is the discovery of the structure of genes in 1977. These are the structure of genes in human cells with their introns and exons and the complexity of splicing, which told us how messenger RNA was made, how it was regulated, and laid out a footprint for further discovery. So both of these happened in 1977, 75, and Biogen was created in 1978, was founded. And Danny Wong, as I will explain, was a member of the scientific advisory board of Biogen and a co-founder. Now, this idea of a scientific advisory board and a co-founder is really something that you can almost trace uniquely back to Biogen, because Biogen was the third company organized. Genentech was Boyer and Cohen, and Thesis was founded by, in biotech was a conversion from a microbial screening company. So Biogen had this organization of an advisory board and a co-founding set. I'm going to talk about that. I'm going to talk about Danny's collaboration with Charles Weissman, which is actually an interesting story about the early days of biotechnology. And I'm going to talk about Danny, not in detail at all, just mention here, that this was in the energy crisis in the late 70s, and Danny led a project of conversion of corn stalks to ethanol that Biogen was supporting at those early days. And that really was, Biogen was founded as a company to bring recombinant technology to all fields, biomedical, chemical, and energy, and ultimately focused on biomedical science. So let's talk a little bit about Biogen Establishment. This was in 1978. Biogen was formed by a group of scientists that held a meeting in Geneva in the early 1978. You'll see down at the lower left, Wally Gilbert and myself. Standing behind us is Ray Schaefer. He was a venture capitalist from Inco, and they had just started a VC activity. And the other venture capitalist who backed Biogen was Kevin Landry at TA Associates. To the right is Dan Adams, who was associated with Inco. Behind Dan Adams is Charles Weissman. Seated at the table is Ken Murray. Moshe Alafi is the second from the left at the top. And this group met in Geneva early in '78. Next meeting was in Paris. The scientists didn't trust the venture capitalists and business people. There was a lot of tension. And ultimately, it was agreed to found the company. This was the date in which the company was officially founded in Zurich at a dinner. And we had a round series of $1 million. That was the founding capital of the company. So shortly after, all of these people are basically molecular biologists. And as we were thinking about what our objective was of how to bring this technology to the marketplace, we realized that we had to have production and processing and the other aspects of a company. So almost immediately, Danny Wong, who's sitting on the left in the back there, was invited to join as a founder and a scientific board member of Biogen. And Danny became an instrumental figure in Biogen. He was the individual, as you can see on the left, the second person there next to Ken Murray. He was instrumental in leading Biogen in production and manufacturing and the whole process of making a relationship to that side of the biotechnology industry. So Danny became a very important figure in Biogen early on, and I will tell you a story. This is Charles Weissman here, looking down at the table. This was something like 1979, 1980, and Charles and Danny collaborated in a very important advance in that time period. Now, Danny continued on the scientific board of Biogen up through 1990. This slide you saw last night. It's a comment after Biogen had begun to become profitable with Avonex to treat multiple sclerosis. And we were focusing on biomedical applications. And Tom Stasso, whose picture he's peeking around the corner, at a scientific board meeting had called all these basic scientists and engineers as propeller heads because they actually didn't understand medicine and didn't have advisory members who were, other than himself, who were deep in medicine. And the next meeting, Vicky Sato, who at that stage was the chief scientific officer at Biogen, at the next scientific advisory board meeting showed up with all these propeller heads and had our picture taken as a set of propeller heads. But Danny Wong is right here to her left, and Wally Gilbert is there to his right. And Danny continued to lead Biogen's efforts in fermentation and bioprocessing. And this shows a celebration of Biogen later when Biogen was profitable. We enjoyed celebrations, so we had several. This was in the late 90s, and there's Danny the third from the left. We're in Zurich at a dinner. Wally is to the right. Moshe Alafi is next to Danny. Kevin is next to him. And we are all celebrating the success of Biogen at that stage. At that same dinner, Danny, and this was with Vicky, and Danny were present, and I really like this slide because every time you saw Danny with Vicky, he was shining. That relationship was extraordinarily important to him, and this is the way I remember Danny. So Biogen started in 1978. Danny was an integral part of it, and I want you to recall how much an impact that had on New England. In 1970s, this is a picture of Kendall Square. There's a Volpe building out there, and Kendall Square is right there. And it's a parking lot, and it's a vacant lot. And though we focus on Kendall Square, New England at this time was in a economic recession. Digital, Wong, Polaroid, all of whom were struggling. The economic growth didn't really have a base in New England at that stage. Manufacturing had gone south. Other activities had changed. And that, over a period of time, due to the leadership of people like Danny, became Kendall Square. And if these curtains had been raisable, you would see out this door 5 major buildings being built by MIT in what is the most economically vibrant area in the country almost. So this technology and leadership by Danny and his technology in biomanufacturing and biopurification allowed this industry to thrive in New England and to actually change New England into a high-tech region where, oh, that's fine. Converting Cambridge and New England from a candy manufacturing into a DNA high-tech community. And that's a product of his leadership and his colleagues who were trained by him in this part in Cambridge. And it's the same story if you look across the country. So Biogen was founded in 1978, as I commented. And if you think back to what 1978 was like, and this is a story about Danny's interaction with Charles Weissman, it was about cartoons or ideas like this. This is a flash garden. It's a cartoon from 1960. There's a patient in a biohazard containment environment who's suffering from infection, and this could be it, interferon. Interferon was discovered in the 60s. It was antiviral, and people had the hope that interferon would suppress the infection and cure the patient, which is what happens here. We still struggle with infectious disease, and interferon has become part of a major treatment for infectious disease. But in the 1970s, you could not obtain interferon in adequate quantities to do any clinical trials, essentially. It was produced by viral stimulated cells, and in low amounts physically, but high amounts in terms of activity. Charles Weissman, as part of Biogen, undertook the project supported by Biogen of cloning human alpha interferon. And this he did at the University of Zurich. And that cloning activity in his lab was led by Nagata, who's now a professor at Osaka. And I'm going to talk about this particular event in the history of Biogen and Danny Wong. So these slides are from Nagata. We were celebrating the 40th anniversary of Biogen, and he showed these slides. And I thought they were very emblematic of the time. So at the stage, Charles had developed a system to try to clone alpha interferon by stimulating cells with a virus, taking those cells because he knew they had induced the interferon genes, and competing RNA with unstimulated cells, and then making competing cDNA, and then taking the cDNA that was not subtracted by the non-stimulated cells, cloning it in plasmids and testing it for interferon activity. At the time, there was no monoclonals to alpha interferon, no purified protein, and the only assay they had was to send the material that they purified as selected RNA to Helsinki to see if it would produce anti-interferon effects in cells. They persisted, and then on Christmas Day, or the day before Christmas in 1979, Nagata in the laboratory was testing individual clones inserted in a PBR22 plasmid for effects if they encoded in bacteria human alpha interferon. And what is shown to the right there is a titration assay where the extract of the cells is inhibiting the virus replication. So the day before Christmas in 1979, they obtained very conclusive evidence that alpha interferon was being produced in bacteria with a cDNA, and that was the first cloning of alpha interferon. At the time, Biogen held its scientific advisory board meeting in the Caribbean. Charles was the first speaker at that meeting. He reported the data for cloning of alpha interferon. He didn't want to have a public announcement until he talked to scientists, and I arranged a seminar for him at noon on Monday at MIT. And he flew with us back to MIT, gave the seminar, we held a press conference. Schering-Plough, who hold the rights to alpha interferon, market value increased a third that day. So Charles was in very large trouble, primarily because he didn't publish the paper before he had the press conference. This is Nagata here to the right. But that got cDNA, but it didn't produce enough protein or enough protein in those bacteria to actually purify alpha interferon and begin clinical trials. Schering-Plough owned alpha interferon, having been licensed from Biogen, and to produce the material that was used by Schering-Plough, Danny and Charles and Biogen got rights to an incubator production facility in Eastern Europe and purified alpha interferon produced there and supplied it to Schering-Plough. But the interesting issue is on the evening in 1981, this was basically 1 year later, Charles, after the cloning, Dr. Weissman called me into his office and said, tomorrow morning, we are shipping the recombinant alpha for clinical trials. I will inject it in my vein, and if I was dead by tomorrow morning, please stop the shipping. He injected 1 million units, wham. He had one of the first major interferon-stimulated side effects. He recovered after hospitalization, and the material was shipped to Schering-Plough, and Schering-Plough had alpha interferon registered in 1986 for treatment of hairy cell leukemia. So Danny and Charles were instrumental in developing the alpha interferon that now still is used under the Merck. So Danny stimulated the whole biotech industry by his engagement in Biogen and other companies his training of the people we've heard about, and was one of the founders of this industry at a time in which it was in its embryonic state. But he also had an enormous effect by his activities at MIT. And that primarily came out of the leadership in forming BPEC. As you know, the NSF established the Engineering Research Center program in '84. Danny was part of an advisory group to that activity. He then led the formation of BPEC. But what is mostly lost in this activity is that this is the first real integration between biological science at MIT and engineering. Because I, Harvey Lodish, and others joined in being on the NSF Center Grant for the foundation of BPEC, and there was number of collaborations that came of this. So this is the first, or at a molecular level, for sure, the first collaboration between biology and engineering at MIT. So what's come of this interface that was nucleated by BPEC? Many things have changed at MIT. The Department of Biological Engineering was established. A third now of all MIT engineering faculty have interest, and many of them have activities in life science. And MIT now has collaborations with essentially every aspect of biological science. If we look from the biology perspective, the modern Department of Biology began in the 50s, late 50s, with the recruitment of Salvador Luria and Boris Margazani. And the Cancer Center was formed in '72. At '74, I came to MIT as part of the Cancer Center. The Whitehead in '83, just as recombinant DNA was exploding across the country, and MIT had begun to think about BPEC. '93, biology joins math, physics, and chemistry as a core requirement, requiring the students at MIT to have a language of molecular, cell, and biological science. Expansion of the neuroscience happened in 2000. Establishment of the Broad and the Human Genome Initiative in 2003, and then the Koch in 2010. So this whole nucleation of engineering from '93 on was a major aspect of MIT's growth and interest in expansion of biology and life science. And this was part of what became a theme at MIT around convergence of engineering and life science. And I think it's best expressed by what Susan Hockfield wrote here, that advances in information technology and material imaging, nanotech, optics, and physical chemistry, coupled with advance in computing and modeling, have transformed physical science. They're now beginning to transform life science. And that is changing MIT and has changed MIT over the past decades. The last institute there, I think, is particularly interesting as an example of convergence, and that's the integration of engineering and biology in the study and advancement and translation of science and breakthroughs into treatment of cancer. And the Koch Institute was established, as was on the other slide, about 2010. It's a very interesting integration of biology and engineering because the faculty of the Koch is about half engineering and half biology. Each floor has engineers and biologists intermingled on it. And there is a number of central activities that bring engineering and biology students together. In fact, today, if I wasn't here, I'd be at a retreat on Cape Cod where the faculty and the student in the Koch Institute are discussing advances in science and sharing ideas. This is the faculty in 210 that established the Koch. And what you'll see up here in the second corner from the right is Paula Hammond. And what you see over there is Angie Belcher, the chair of chemical engineering and the chair of biological engineering. So this group, the cohabitation of biologists and engineers, has been tremendously successful. It has been ranked the #1 basic science or cancer institute in the country, non-clinical cancer institute in the country, and it is integrating biology and life science in a way that no other campus is doing. And it has continued to help transform New England. And we can trace this back almost linearly to BPEC, Danny's leadership in 1985. So the legacy of Danny Wong is much bigger than this wonderful group of people who've come together to celebrate being in his lab and being part of the biotechnology, fermentation, and bioprocessing community. Obviously, the major legacy is his friends, family, and students, but we also should think of Danny in this period as being integral to the growth of biotech, the transformation of Kendall Square, Massachusetts, New England, and the country, and the integration of modern life science and engineering at MIT. Danny has changed through his leadership, charm, and determination, the history of MIT, and changed in many ways the history of life science and healthcare in the world. So he was a great friend. I loved his Chinese food. I loved his can-do attitude. I loved his kiss, keep it simple. And I just enjoyed being a colleague with him. Thank you.
To the schedule for questions, especially to be able to ask those and I will start the questions to give others a chance to think. I think you laid out beautifully how, in the story of Biogen, this integration of life sciences, fundamental sciences, and engineering was really critical towards seeing the translational results that we've seen. And you also pointed out that at MIT, we now have a third of all faculty who have some affiliation with life sciences. And the scope has grown to be much larger than human health. So what are the lessons that we can learn to deal with climate change, for example? Or when we start talking about resilience, we need to be able to both understand biology, perhaps manipulate biology, now in a different realm that's talking about the health of the planet, for example, as opposed to the health of the body. What can we learn, and what should we be doing?
Well, at MIT, we can contribute in multiple ways. We are. I mean, the energy program is a major contribution to what is going on. The whole conservation and alternative sustainable energy sources. Life science is still contributing to bioprocessing, to other sources of hydrocarbon and manufacturing. and removing hydrocarbon-based materials from our economy. So everything plastic is basically hydrocarbon. It's very difficult to degrade, and it's a pollution in real world. We should have biodegradable material. We should expand that. But what I really want to emphasize is that this community has to look forward and make major changes. Converting energy from hydrocarbon to other sources is not going to be a 2-year affair or a 10-year affair. It's going to be decades, and we need new sources of energy. And therefore, one of the things going on at MIT I find not being emphasized even on this campus as much as it should be is fusion. And I think alternative energy sources that do not generate waste like fission and nuclear reactors as we now have, in the long term, could be extraordinarily important to societies. So I'm just saying we have to remain the most innovative, integrated science, engineering, community university as we can be, because we transform the economy and the world out of life science. We've done it out of computing, but we're going to have to now do it in engineering, in energy. Charlie.
Phil, you very nicely described the evolution of this integration of biology, engineering, chemistry, multidisciplinary, and we've made a lot of progress. But to deliver on the vision that you just described, what do we need to do in educating our students and our postdocs and others in order to achieve that vision?
Well, I think one of the most important things, and that's why I talked about conversion and the integration of life science and engineering, is we have to have students who talk both languages, who can learn both in the life science community, the biomedical community, and the engineering community. And that happens best when students are together. And all this masking and restrictions is a real handicap that'll pass. But bringing students together and fellows together in an environment where they can share is an enormously important thing. And what we're going to also have to do at MIT is the biggest issue, in my opinion, in biotech and pharmaceuticals is the cost of the process, the cost of healthcare. So how do we impact on the cost of healthcare and still keep innovation in the system? And we have to bring technology of identifying patients who will benefit from a given treatment early in the process to get a major therapeutic effect and reduce the cost of ultimately healthcare. So we're going to have to integrate our education in early diagnosis, healthcare delivery, communication, and participation of patients. So I think that's a major frontier that MIT is developing communications with. I'm very excited about the embryonic state of AI to life science and happening on the campus. And I think it could be extraordinarily important.
So it's interesting to hear that Biogen had among its initial potential targets this energy orientation. And indeed, many of us were there, as students working on the Department of Energy funding for the very same reasons. So I wonder if you could say more about how rapidly Biogen transitioned and understood that that wasn't a logical direction. Many of us made that change as well.
So in the late 70s, early 80s, Biogen, with the Scientific Advisory Board, each individual had developed, on the board, developed a project that they had expertise in. In Danny's, it was in bioconversion. And we also had a major relationship with Monsanto in chemical, productions in chemical. And we went out to try to sell deals, to try to find an opportunity where we could reduce the cost of a commodity, such as ethanol or other intermediate chemicals, by bioconversion type processes sufficiently to gain value that would sustain the development of the company. And it turned out that when with a cold view, that was not possible at that stage. And in biomedical science, where there was an enormous margin for new technology, that became the focus. And clearly Genentech was the first company established, and they immediately focused exclusively on biomedical science with human insulin and growth hormone, and et cetera. So after testing it in the marketplace, there was this conversion to biomedical exclusive in Biogen.
Jamie's question, at least, like Jamie, I started out here working on bioconversion and actually went back to doing it again 10 years ago when it was no longer a question of price, but it was a question of carbon and national security and all that. But at least what I've gotten out of it is that at the end of the day, if you're going to do kind of high-end innovation in a commodity, that's very, very hard because the commodity price, the fungibility of a commodity means that it will always be at its lowest possible price. Until and unless renewable energy is no longer a commodity and is actually priced as a specialty product which is valued, I don't think, the promise, the circular argument, there will be no innovation that can actually last that long to be able to be affordable for a commodity. So we backed away after $100 million spent on it because at the end of the day, you can always go work on a specialty like pharmaceuticals, like even agriculture, where the price can reflect value. In the energy sector, it's the fungible commodity.
I think totally agree with you. I think the interesting story is one that you have referred to, and that is the development of technology in the plant industry. Because there, the life science revolution had direct implications for what could happen. And because of the political response to genetic engineering, the lack of acceptance internationally, the local resistance to recombinant DNA, it's amazing. People will go inject products for recombinant DNA into their veins, and they're concerned about some little gene floating around in a plant. It's just amazing. But the societal acceptance of this technology is really essential for it to be successful and to grow. And that's my concern about, biomedical technology, that if society's acceptance of this innovation in constraining cost becomes sufficiently negative, it could seriously compromise innovation in this industry and for the patients.
So generics show.
Yeah.
If they are able to price an innovation as a generic, it will make it a commodity. There's no way you can innovate that.
Bill, in the last 40 years, we saw a lot of the computing and electronics companies move west, and a lot of the life sciences companies move east. What drove that, especially in life sciences here? Why did this, you could argue early on the West Coast had an advantage with life sciences companies, and now it seems, when we're looking outside, there's a tremendous amount here. Was that economic policy? Was that?
No. I mean, Massachusetts has become more economically supportive of high tech over the last 30 years. but it wasn't economic policy. It was the integration of the discovery part of life science with the medical community and the translational part. Our hospitals contributed enormously to this revolution in New England. And because we were across the street from Mass General and the Brigham and Dana-Farber and the Children's are all number 1, 2, or 3 in the country as major hospitals with major research programs. So this community was attractive for making that interaction. It was attractive for bringing people to this extraordinarily vibrant, learning, and innovative community. So it just grew, whereas the more separated activities on the West Coast, where 30 miles from the nearest hospital or nearest biomedical or medical translational environment, it's more difficult. And it's also embedded in a high-tech community that has its own objectives. So venture here and financing and communication is focused in biomedical technology. On the West Coast, it's IT and social media. So I think they segregated because lack joins lack, and that's been a big part of this.
There is one.
I have a lot of questions. So I think, the part where we all kind of leave it off as though it's non-scientific in our discussions is regulation. And it's almost like, well, that's beyond our pay grade. And yet, the more convergence, the more advances, the more we're going to generate a massive pileup at the gates of regulation with an inability to actually advance these things. We've gone through a period of 18 months where the FDA had to act with a true mission of risk-reward instead of no risk and maybe reward. But yet new technologies are always going to introduce new risks. So how do you feel the academic world, the scientific world, can actually dare to go into that space with the same type of innovative mentality, which in the last 35 years I've observed, we generally tend not to do because we think you're not supposed to mess with that because that's just that's the police force almost. And yet, without those reforms, I don't know how any of these synthetic biology, disease responsive, drug disposing, none of that can go forward.
I think the one aspect of that is to continue to support more innovation and science in the FDA. So the more that the FDA keeps abreast of what the technology is able to produce and what the benefits are, the better they will be at making this gatekeeping decision. And the FDA is under enormous political pressure, as you know, from watching what's happened over many decades. So it's where politics and innovation mix in a very important point in this whole technology. But I do think if we can move technology into the primary care office and then in the system to identify disease early and show we're creating real value in changing the course of people's health and dealing with inequality in that issue, I think is a very important thing for us to do as an industry here, is going to be essential to get political support for this regulatory, necessary regulatory activity for the industry to thrive. Thank you.
So good morning, everyone. I am standing in, as I said, temporarily for Paula Hammond. My name is Chris Prather. I'm the Executive Officer in the Department of Chemical Engineering. All of you alumni know that that's just a fancy name for the Associate Department Head. And so I am standing in now to introduce our next speaker, who I could say needs no introduction because everyone spoke so much about how much they appreciated their big brother yesterday and the constant companion to Danny in research and teaching in the form of Charlie Cooney. Nevertheless, I have words on a page I am going to read so that the report can go back that I did my duty as I was instructed to do. So, Charles Cooney is the Robert T. Haslam Professor Emeritus of Chemical Engineering at MIT. He did his graduate studies in what was then the Department of Nutrition and Food Technology, where he was one of the first advisees of Danny Wong. After working briefly at the Squibb Institute for Medical Research, he joined the faculty of MIT as an assistant professor in 1970. His research and teaching have spanned many aspects of biochemical engineering and pharmaceutical manufacturing, and, as we know, he was a longtime collaborator of Dan's. His awards have included the Gold Medal of the Institute of Biotechnological Studies in London, the Food, Pharmaceutical, and Bioengineering Award from AICHE, that's Division 15 for those of you in the know, an election to the American Institute of Medical and Biochemical Engineers, among others. From 2002 to 2014, he was the founding faculty director of the Deshpande Center for Technological Innovation. So a lot of what you heard Phil Sharp talk about in terms of this ability to translate technology from MIT into the commercial space, Charlie's played a really foundational and phenomenal role, both here at MIT and around the world, in pushing that forward and providing support mechanisms for researchers and academic institutions to do that translational work. Charlie is also a consultant to multiple biotech and pharmaceutical companies. He sits on multiple public and private company boards, and his past public board positions include those at Enzyme and PAL Corporation. Please join me in welcoming Professor Charlie Cooney.
Thank you, Chris. I would prefer to be the older brother and not the big brother, which has other connotations. But I do cherish that reflection. Putting this presentation together was actually quite a bit of work. Yes, I've resurrected some slides that many of you have seen before, but I've put a new twist on them. Yes, I have reflected back on the journey that I have had here at MIT, and I've tried to, well, not tried, it was actually quite fun to resurrect some stories, some memories that some of you may know, some of you may not know, and I think some will be new. And at the same time, what I would like to do is to reflect that I'm looking at the world through a lens that Dan taught me to look through. I have embellished it, I have expanded it with the assistance of everyone in this room. And it's that story that I'd like to lay out, but I also want to finish with some thoughts on where we're going to go. As Dan would always never hesitate to tell us. That's different than telling us where you should go, but where we want to go, and that's the important lesson. So going right back to the beginning, this is about building a foundation on fundamentals. All of us have heard Dan expel on that at great length, often at times during seminars when we wished he didn't. And it's the truth. We do need to focus on the fundamentals. So these are reflections on the future. And let's see where this takes us. As soon as I find the right button to push. Yeah. Okay. So where do we begin? First principles. So the first principles for this morning are that we are here to remember and celebrate his life and what he taught us. Second, we are not only the beneficiaries of what we have learned, but we're the agents. You are the agents of that legacy, and it will continue. You are delivering on the vision. We'll hear more of that. We heard it yesterday, but we'll hear a lot more this afternoon as we look deeply at the future. So I've seen the hard work. I've seen the tears. I've seen the frustration leading to your successes. And most important, I have seen the ability that all of you represent in being able to solve important problems and to share those solutions with others. And that's what we learned. And once a student of Dan's, always a student of Dan, and I will share you some of this 55-year journey as a perpetual student. As I said last night, many of you had the benefit of graduation. Some of us never have. But, you know, we can aspire to that as an endpoint, perhaps. So let's go back, and I'm going to share a few slides that some of you have seen before. Newbar and I published a paper. Doug Cameron, who's here and has a hobby of genealogy, put this particular slide together, published in 2006. And what you see is an amazing legacy coming back from Dan, going from the top to the bottom, down to 1453. And the ability to trace that legacy and the mentorship that has transpired over 500 years. But as I will warn you that I will inflict on you some of my hobbies, one of which of my hobbies is cartography from the 15th, 16th, 17th century. So I decided to look at the beginnings of this legacy and ask, well, what did the world look like to people at that time? And it's rather interesting. Well, first of all, the maps that come from the 1500s, so the 15th century and even the early 16th century, are based on the coordinates of Ptolemy, which is 2nd century, Claudius Ptolemy. And those coordinates were developed by travelers. And this is before frequent flyer mileage. Those travelers reported back, and Ptolemy collected this information, and European cartographers from the 15th century began to codify it onto a map. And so if you look at this map, where are we? Where are we here? Well, this is the Malay Peninsula. And you'll notice that the Indian Ocean, if you could see the whole full set of maps, is actually enclosed. And when you begin to think, and here's India over here, and parts of China are up here. So the world was very different. Well, the world wasn't different. It just, they thought it was what it was. And one of my favorite maps in this small collection that I have is this one. So we jump forward 50 years. This is a map by Sebastian Munster, 1550. And you begin to see some things that are more recognizable by the way we know the world today. Here we have, whoops. Okay, here we have the Malay Peninsula. Here we have India. And we see that the Indian Ocean is no longer landlocked. I guess that was climate change. Somehow Japan seems to have gotten lost. Australia, I'll let Australia speak for itself at in this map. This is a 500-year-old manuscript map, and to hold it is just inspiring. But I fell in love with these maps for two reasons. Okay, this is taking us back to the early days of our legacy of what the world looked like. And the reasons I fell in love with these maps is they're beautiful works of art. They're hand-painted. They're made by wood. This was made by a woodcut. But the really interesting part of the maps is they're wrong. They're wrong. They're not wrong because the cartographer wanted to make them wrong. This is the state of knowledge at the time. So every time I look at these old maps, I become very humble. I realize what we don't know or what we didn't know. And of course, all of us who have walked into Dan's office have been made humble by what we don't know. And it was that appreciation that I developed in that early history of working with Dan that really led me to appreciate these very early maps. And despite their beauty, and despite what we look at and the inspiration, they're wrong. And they're wrong because that's the state of knowledge at the time. So we look at the tree. You've seen this. You'll see more of it, I'm sure, in some other slides. That was put together in 2006, celebrating Dan's birthday. And what I want to emphasize is not the tree and the beautiful shape of it, but this tree is alive. This tree is growing. And when we look at another way of displaying Dan's legacy, it's the dynamic impact of 5 decades of teaching and mentoring that keeps this tree alive. That's what it's fertilized with, and that's what it's watered with. And all of us, and many of you in the room are educators, are really expanding this. But even those of you who are coming from industry are expanding this legacy. We are the agents of the legacy that Dan laid before us. This happens to be the academic tree. We can also see, of course, these are unreadable. This, if Dan saw me show a slide that was this unreadable, you know what he would say. But sadly, he can't. But it displays the dynamic impact in academia and industry that continues to flourish coming from that legacy. Now, Matt Crokin showed this slide yesterday, which is a map of Dan's publications across multiple areas of research, and of course, over 5 decades. But the point that I want to pick up on is actually the point that Phil made earlier. And that what Dan taught us to do, what he demanded that we do, what he actively did, was to bring people together to create a community. So when you look at all of these papers and you look at the areas of research that they represent, and the fact that they continue largely across a large span in time, it was really about multidisciplinary collaboration. And that's how you address important problems. And that legacy remains with us. So let me step back and share my early journey and my introduction to Dan, because it spans 55 years. And it began really with my being an undergraduate at Penn with Art Humphrey. And those of you that heard the conversation that Art and I had yesterday could appreciate some of that. Now, I arrived at Penn in 1962, which was when Dan left. So he didn't really leave. His legacy was there. People talked about him all the time. His dirty glassware was there, and I had a chance to wash it after he left. If you're going to do a PhD thesis in 2 years, you leave your dirty glassware behind. Well, anyway, I learned how to wash glassware. And I wasn't until 1965 when Art suggested that, by the way, I had no interest in going on for graduate work in those early days. I was having too good of a time as an undergraduate. But fortunately, I edited those parts of the conversation out of the version that you saw yesterday. But nonetheless, I became involved in undergraduate research, the equivalent of Europe at that time. And I became inspired. I became intrigued. I became excited by doing research. And it was that inspiration with Art's guidance that led me to an introduction to Dan. Why don't I go to MIT? So I made the journey. How many of you remember Mohawk Airlines? Good for you, Gene. This is Mohawk. I flew to Boston. It was the second time I had been in an airplane in 1965. There was no frequent flyer miles. I think we had seat belts. There certainly was no security. And I had an interview with Dan. He was on the building 16, second floor, and his office was buried in the back of a laboratory. I found it quite expediently. But after the interview, I proceeded to get lost for the first time, many times followed, in the infinite corridor. You know, you need to learn to make the lefts and the rights in the right sequence, or you don't end up where you want to end up, which was my story. But that, on that interview, I was successful. I was accepted into the Department of Nutrition and Food Science. And I began working with Dan on a thesis. My lab bench was right outside of Dan's office. I couldn't hide. Good news was, if I had a question, Dr. Wong, I could catch him on the way in or the way out. Bad news was, if I wasn't there, it was noticed. But I survived that. And then, of course, like many of you, this was my first introduction to chopsticks. The other graduate students, fortunately, a couple of whom were Chinese, introduced me to the use of chopsticks. And that turned out to be very important. Now, the good news is that I also was introduced to the best Peking duck coming out of Vicky's kitchen that you could ever imagine. And I became spoiled on Peking duck. And this began very early in my career. But then some things that maybe you don't appreciate. Money was tight in those days. We didn't have these big grants. Biochemical engineering was not funded by many of the government agencies. We didn't have industrial funding. Fortunately, we did have a training grant. I'll talk about that in a moment. And we could literally scrape along. So when we needed equipment, Dan and I would drive down to Taunton. It's about 45 minutes south. And there was a government warehouse where everything that the government didn't want, much of it vintage World War II and Korean War, would be shoved into this very cold, unheated warehouse. And we went there every month, 12 months out of the year. And we would retrieve recorders. We would retrieve glassware. We would retrieve instrumentation. We would retrieve cool stuff that looked like it might be useful sometime. And we would negotiate and pay a small amount of money, load it into Dan's car, and drive back to the lab and unload it. And that's how we equipped the lab. Also in those days, when I was a graduate student, I had to buy my own glassware. So I went to the lab supplies at MIT, and I put it on my personal account. I was very careful with my glassware, because at the end of my research, I would take it back. And the good news was that between when I started on my research and when I returned it to lab supplies, the price went up. And I could graciously say, well, they would say, I can't give you any money back. I said, that's okay. I'll be glad to get my money back, even though I had broken a substantial amount of glassware. So these were frugal times in terms of what we got, how we used it, and how we treated it. Life got better. And then I finished my master's thesis. So I started in September of '66, and I finished my master's thesis in June of '67. And I was going to go on and start my PhD thesis with Dick Matalis. Well, just as I had agreed to do a PhD thesis with Dick Matalis, Dick said, by the way, I'm going to go on sabbatical for 2 years to Israel. Okay. That's good, I guess. It turned out to be terrific because I didn't have an advisor. And you can make a lot more progress in your research when you don't have an advisor. But the good news was, and the really fortunate opportunity for me, for which I am eternally grateful, is that Dan became my advisor. He had been my master's thesis advisor, and now he was my PhD thesis advisor, and I worked with him. Occasionally, I get a little message from Dick Matalis long after I had solved the problem that I had, and it was under Dan's guidance that I learned to solve those problems. So that was the opportunity of a lifetime. Now, of course, in the beginning, it was Dr. Wong. I eventually learned to call him Dan. I never called him Danny. It was always Dan. And to my sons, Matt and Brendan, who many of you know, it was Uncle Dan. So here's a man with many shades and forms in his legacy over a family. My master's thesis, let me indulge in this for a moment because this was important to where we were in biochemical engineering, and it was important in the way that you can see how Dan was thinking. I went into his office and said, what shall I do for my master's thesis? And he said, there's this problem that heat of fermentation is actually a cause of failure as you scale up to large reactors. And we don't have a good way of predicting what it's going to be. And as many of you heard him say to you, go figure it out. And I went to the lab, and that's what we did. You can see we haven't changed in our pictures in all those years. So you can't finish till you start. And this memo, my first progress report to Dan in November 1st, 1966, is typed with a real typewriter. And if you look closely at the E and the A, you can see that I didn't know how to clean the keys on my typewriter. We ended up with a very simple correlation. After running many fermentations under many conditions, heat of fermentation is equal to 0.12 times the oxygen uptake rate in millimoles per liter per hour. And that turned out to be fundamentally correct, experimentally verifiable, and the basis of a master's thesis, and the answer to Dan's question: can you take an industrially important problem and come up with a way of predicting the answer? And the answer turned out to be quite simple. So there are tactical lessons that we learned, and these are the tactical elements of Dan's strategy. Build on first principles, understand, build the foundation, have a vision. Maintain your values, the pride he had in his students, the humbleness, the gentleness, although he didn't always seem gentle, as we all know, and the mantra, I can do it. So let's step back and look at the historical landscape in which he and we have grown up. So biochemical engineering in the 40s was solvents and chemicals, very critical to the war effort. 1950s, it became antibiotics. the challenges of scale-up and aseptic processing. In the 60s, it was about expansion of products and the promise of continuous culture. Yes, fundamentally beginning in the 50s and the 60s. In the 70s, it was expansion further to food, single-cell protein, biological products from renewable resources, harnessing enzyme technology and enzyme engineering, and building on this foundation of synthetic bio that Phil spoke to a few moments ago. And then those exciting days as we get to the 80s and through the 90s and to today, when we built on that foundation of synthetic biology, increasing complexity, and the demand that we have going forward, delivering these benefits to society. And that's your responsibility. And that's the responsibility of all those who are working with us, our kids and our grandchildren. Let me build a bit further on the theme that Phil spoke to a moment ago. And that was really Dan's leadership in shaping education and research. And the operative word is shaping. He didn't do it alone. He did it with all of us and many other colleagues that we all know and have worked with over the years. In the 1960s, he was successful in getting an NIH training grant in biochemical engineering based upon the principles of teaching chemical engineering and biology as part of a curriculum and supporting research in that area. I was supported by one of those training grants. The course that Dick Matalis started but quickly abandoned, and Dan really picked up in the late 60s, fermentation technology, and continues to this day. And you'll hear more about that. But it was important because what it did was to take science from research in the laboratory into industrial hands and to listen to the people in industry and bring their problems back into the lab. It was a two-way street, and we got paid for it. So it was a good deal for all. The 1970s were really quite pivotal in shaping education and research in biochemical engineering. The Sloan Foundation was supporting a major project at MIT on single-cell protein. The problem was, how were we going to feed the world? And many of us did our research with the mindset that we were trying to convert hydrocarbons, methanol, and alkanes in order to produce protein for animal and human food. Now, we were very successful technically, but something we didn't anticipate. The price of food went down, the price of oil went up, and the technology was a resounding success, but the business case, to put it into precise technical terms, sucked. And it didn't work. But it was on the foundation of that we brought together people from multiple disciplines, began to bring them together to work on solving an important global problem. National Science Foundation Enzyme Engineering Grant. I remember going to Washington with Dan early 1970s. We had this vision, a group of us, included George Whitesides, Tony Sinsky, and others, that we could take enzymes and harness them and pull them together and use them in a synthetic way. Synthetic biology, as Phil described. And we went to NSF with this proposal, and we had our slides. Now, slides. Do you know what slides looked like in those days? They were lantern slides. They were this big. They were made with Polaroid film and mounted in a piece of plastic and put into a projector with a very powerful bright light that would shine the image on the screen. Dan and I get to the pivotal point at NSF, and all of a sudden we see a black dot on the screen. It begins to expand, and smoke is coming out of the projector. Well, we put the fire out and proceeded along with the talk. But that was the first time that I learned it's good to have slides, but be prepared to give the lecture if all things fail. That very successful program that began to bring people together. At the same period of time, the example that Phil spoke to, the Department of Energy funded a very major project in the late 70s on the conversion of cellulosic biomass to fuels and chemicals. Many of the same things that are being funded today were funded then. The only thing that's different is the technology today is much more sophisticated and the solutions are much more practical than they were then. But we learned a lot. That's when there was a publication of the book, Fermentation and Enzyme Technology, that set the teaching criteria and paradigm that has lasted over decades. And then it was in the 1980s, as Phil very eloquently described, this very pivotal time when we formed BPEC. And we brought together biology, chemistry, chemical engineering, and we brought it together not just in the research laboratory, but we brought it together in the classroom. And we learned that important lesson. Faculty who teach together do research together. And it's about bringing the students in the same place at the same time. And they walk between the laboratories in a very seamless way. A very exciting time that many of you sitting in this audience were part of. So what have I learned in 55 years? Well, why? Big lesson I learned is that manufacturing sits on the critical path between science and the patient and society. What the interesting and important things happen at interfaces, and how you need to venture to uncomfortable places, take risk, innovate, and learn how to ask those unlocking questions. Now, one of my hobbies, Dan never approved of this, but I did it anyway. It was high altitude mountaineering, and some of you have seen these slides before. But the message is that when you embark on a journey of developing a new product, developing a new process, developing a new company, you have goals and aspirations. In this particular case, the aspiration was to climb this mountain, Amadou Blanc, and you can see that it sits in the shadow of Everest, in the Khumbu, just south of the northern Nepal, just south of Everest. And when you embark on this journey, when you go into the lab, when you walk into Dan's office, you're taught to recognize that there's uncertainty and to embrace risk. And if you didn't think it before you walked into the office, you certainly embraced it when you left. And yes, that as you stand there and think, this is where I am, and that's where I want to go, and it's several days away, there is some associated risk. And I'm standing here behind my son, Matt, who's on the small ledge that looks like a dead end. It is, because the only way out of there is up. And you're doing your experiments a step at a time. So this is no different than the way that all of us have behaved in the laboratory and in our company startups and in the life that we have lived. It's thinking about how you take that step and how you take it cautiously, but being willing to take the risk. And this is a photograph of my son, Matt. This is it's not that difficult of a rock climb, except that it's at 20,000 foot altitude. You have a 40-pound pack on your back, and the distance from the yellow boot to the ground is about 2,500 feet. So what you learn is that it's an entrepreneurial journey does have risk. And what Dan taught us, yes, Dan taught us this fact. I was thinking about it at this very moment when I took this picture. The impact is something that is, you fail, you fail. It doesn't work. In this particular case, the impact is a 2,500-foot fall onto granite. Not good. Therefore, you need to learn where to focus. And where you focus is not on the right button. Where you focus is on the probability. And that's the lesson that we learned in this journey. It's about the probability of managing the risk. And it's about thinking where you want to go and for the right reasons. So I stand here looking at my feet, and why am I looking at my feet? Why am I not looking forward? Because my goal is to get back down. My goal is not to summit. My aspiration is to summit. But I want to know that when I put my foot down, it's in the right spot, because the downside's not very pretty. It's also 5 breaths per step, so I have time on my hands. But I'm really focused on what it is that's important. And that's a lesson that we all have learned. What is important? So what? Why are you doing it, and what do you desire as an outcome? My desired outcome is to get back down. And if I happen to summit along the way, that would be nice. So when we think about this in the context of what many of us do in pharmaceutical manufacturing, our goal is the sustained delivery of a quality, safe, and efficacious product to the patient. It's about delivery of the product to the patient. That's the outcome. That's the goal. It's not about the pilot plant. It's not about the successful experiment. It's how you translate that to society and how you can do so in a way that can be sustained over a substantial period of time. Now, many of you have participated and have really driven the development under Dan's guidance of animal cell culture and learning how to make monoclonal antibodies. And of course, that has become the platform for making a wide variety of products. And because it's a platform, and with your contributions, it's continued to improve. But that's not where we're going. Where we're going is to a different place, where we have new products. We have new therapeutic modalities that, in fact, are delivering the promise of biotechnology to society, to the patient. And do we know how to get there? It's about embracing the complexity. It's about embracing the biology. It's about going to a place that others haven't been, and it's about doing things that haven't been done before. And I know if Dan were in the laboratory today, this is what he would be doing. Why do I say that with such assurance? Because all of you who are students of Dan, that's what you're doing. And that's the legacy that you're taking forward based on that foundation. We were caught unawares by SARS-CoV-2 and COVID-19. Our preparedness we were sorely unprepared. And as we think about where we've come from these past 2 years, we've come from a former normal. Here we are today. And of course, we're all being asked, when are we going back to normal? We're not. We're not going to go back to the normal. We're going to a different place. And that's the opportunity. That's the opportunity to the new normal. And we need to imagine the industry that is going to take us there. So we seek a framework for advanced manufacturing. We seek a framework to take us to that new normal. And why now? And these are the questions that all of you have gone through in your work with Dan. And you begin to think about these questions in terms of, well, how am I going to do it? And it's through multiple lenses of the technology, of regulatory, of business. And it's through those multiple lenses coming together, it's those interfaces that are required to address the important problems. And as Phil shared with us a moment ago, and as we think forward, it's your ability to bring science all at the same time to address those interfaces that's going to make a difference. It's done through, and this is what we all began years ago, intensification and integration. In fact, it might be the fact that we're going to get to a place that's continuous end-to-end manufacturing, but that's not what we're doing. We may have an aspiration to do continuous end-to-end manufacturing, but what we're doing is we think about these new technologies and these new problems is to really think about intensification and integration. Whether you're an upstream person and thinking about how to improve the upstream, or whether you're a downstream person and thinking about how to capture and purify that product to make it safe and efficacious for the consumer, for society, and in many cases for the patient. It doesn't matter. It's that mentality of thinking, how do I make it better? What's the so what? The so what is taking us to a new place, to a place that we need to be to deliver science to society. And that's what we learned to do. And it's interesting, as I have looked at across the landscape of how multiple companies named have thought about delivering that new process science at the heart of biochemical engineering in making together these processes. The tactics that have been employed, whether it's N-1 to enhance inoculum development, whether it's perfusion culture, whether it's continuous capture or continuous downstream processing, there's no single answer. That's because everybody has a different problem. And that the problem, of course, is defined by what is the right reason for doing it, the why, the so what. What's the business case? And when you identify that and you know what the problem is, only then can you put together the solution. So a mantra that many of you have heard me say many times, that if you don't know where you're going and you don't have a means of measuring where you are, you won't know that you've arrived. And remember back on that slide of Dan's publications over a long period of time. One of those areas of work was analytics and the need to measure and the number of times that we have all been told that are you measuring the right thing in the right way at the right time? And if you don't, you won't know that you have arrived. So in these last couple of slides, I want to leave you with a message that we need to prepare for the new normal. That's our responsibility today, because we're not going back to where we were. We're going to a new place. We're going to a new place in terms of the kinds of products that are going to be delivered. And those products are emerging out of amazing science. We're going to the ability to deliver vaccines and therapeutics globally, not to the 500 million people who can afford it, but to the 5 billion people who need it. We're going to align digital health, getting drugs and therapies that last mile to the patient. And we're going to use a variety of technologies. We need to think about it as building the full stack, end-to-end, of technologies that are needed to deliver science to the patient. That's what it's about. And we need to learn how to connect the dots. And as we think about preparing for the new normal, it's really about delivery of the new normal. And it's that delivery that requires people. You, your students, your colleagues, those who you mentor, and those who you haven't hired yet, but you're going to hire and teach how to do it. And we're now working in a new model, a new workplace. We're thinking about new organizations that are end-to-end, not silos of upstream and downstream, not silos of analytics and regulatory affairs, but of a seamless organization that will deliver that value. And the processes that are required in order to implement that with the people that we train and we educate, and in a business model that's fit for purpose. Not thinking about how we minimize the cost of a unit operation or even minimize the cost of a product, but thinking about it as we deliver the product to the user. And that's a very different business model than the traditional one. We're going to a new place. So when you reach the summit, which my son Matt and I did, we had a beautiful view of Everest in the background, and we were successful in reaching the summit. But that was only our aspiration. And the goal we have is not building that plant. The goal is not doing that experiment. The goal is not establishing the proof of concept. The goal is delivering that science to society and doing so in a sustainable way. When you're on the summit of a big mountain, it's 23,000 feet, 7,000 meters, 75% of the accidents happen on the way down. You're tired, gravity is not in your favor, and it's tough. And likewise, once you have that pilot plant working and that proof of concept established, you're only 25% there. So we need to think about it from end to end and what our goal really is, which, as Dan would say when he asks, so what? The so what is delivering the science that is emerging so rapidly to society. And that's what we were trained to do. And one of these days, I may even get out of being a student, but I hope not, into the rest of the world. Dan, thank you very much. And all of those of us who are students of Dan, thank you. And not just for what you have done, but what you are going to do tomorrow. Thanks all.